طيب السلام عليكم ورحمة الله. We are going to discuss regulation of enzyme activity, and first we are going to state some important information. The type of reactions that are subject to regulation are known as critical reaction or essential reactions. Not all enzyme catalyzed reactions are subject to regulation. When we say critical essential step in a reaction, we mean that this step either irreversible committed step or rate limiting step. When we say committed step, that means it is the step that lock intermediates in certain direction. بمعنى يعني دي الخطوة of no turn back، الخطوة اللي بيخش بها الانترميديت بعد في direction معين to produce a specific product، ودي بيقول عليها committed step، وبالضرورة it is irreversible step. And when we say rate limiting step اللي هي دي الثانية rate limiting step, we mean it is the slowest step in a pathway or is a step that determines the rate of the pathway، اللي هي الخطوة اللي بيتم فيها التحكم في الريت، اللي هي الخطوة الأبطأ عن طريقة we can increase or decrease the rate of the pathway. So these two type of steps, committed step or rate limiting step, are subject to regulation. Okay. طيب, we have different mechanism for regulation of enzyme activity, and these different mechanisms operate within different time scales, some known as short-term mechanisms, some known as long-term mechanisms, and also act by controlling different issues. Some control the distribution of enzyme either within tissue or within the cell. Some control the concentration of the enzyme molecules either by increasing or decreasing this concentration, and some control the activity of the existing enzymes either by increasing the activity or decreasing the activity. Generally, we can say this mechanism include compartmentation, induction and repression mechanisms, allosteric regulation mechanisms, and covalent regulation mechanisms. And also we have multiple type of enzyme inhibition, product inhibition, feedback inhibition, and so on, and this can be also considered as regulatory mechanism of enzyme activity. Regarding compartmentation, it is the distribution of pathways in different tissue or in different organelle. When we talk about tissue, we say tissue specification, and this is an integrative level. It is a level of integration of metabolism. Different pathways are distributed in different tissue. So we can find a synthetic pathway in a tissue and the degradative pathway in another tissue. This prevents competition between shared molecules. For example, if a reaction produces fatty acid, the other reaction which consumes this fatty acid occur in another site, either inside our body or inside the cell. And this is compartmentation. Look here, lipogenesis, production of fatty acid occur in the cytosol, but oxidation of fatty acid through beta oxidation occur in the mitochondria. So this prevents competition. Not all fatty acid produced by lipogenesis is consumed by beta oxidation. If we are going to oxidize the fatty acid, we transport it to the mitochondria for oxidation. So this allows proper control of reactions. The other type of mechanism is control of the concentration of enzymes, and this occur either by induction and repression at the level of the gene, or degradation at the level of the protein. The concentration of enzymes can be regulated by change the rate of synthesis, either increase synthesis, which is known as induction, or decrease synthesis, which is known as repression, or by control the rate of degradation by proteolytic or proteolysis. Okay. This induction and repression mechanism is slow. It takes much time, sometimes hours, days, or even weeks. So it is long-term mechanism, and sometimes it is known as adaptation mechanism. For example, لأنها بتحصل في الليفل بتاع الجين، يعني لما نحتاج مثلا نظبط اكتيفيتي بتاع انزيم معين، بنحتاج نتحكم في انه نزيد التصنيع بتاعه مثلا او نقلله، بتاخد زمن يعني. it is adaptive mechanisms. Allosteric regulation is control the enzyme activity by binding of an allosteric effectors. So what do you mean by allosteric? Allo mean other and steric mean space. So when we say allosteric, we mean other site for binding. Okay, this is an enzyme. The enzyme has active site for binding of the substrate, but some enzymes have allosteric site for binding of the effector. So this is allosteric enzyme. An allosteric enzyme is subjected to allosteric modification. So allosteric enzyme is an enzyme that contain, in addition to its active site, another site. It is allosteric site. This allosteric site is a site for binding of a non-substrate molecule, which is allosteric effector, and this molecule influences the activity of the enzyme after binding. Okay, so this allosteric regulation similar to the non-competitive inhibition, but differ in some ways. Okay, generally allosteric regulation occur for committed steps or rate limiting steps. So any enzyme catalyze a rate limiting step or catalyze a committed step, by default it is an allosteric enzyme. Allosteric enzymes don't obey mechanism-maintaining kinetics. They have a non-mechanism-maintaining kinetics and show sigmoid curve. Okay, and they also show something known as cooperativity. Cooperativity, that mean binding of the effector facilitates sometimes binding of other molecules and so on. So allosteric effector, they are the molecules that bind to the enzyme. Small regulatory molecule bind to allosteric site in an allosteric enzyme, and they have no or little structural resemblance to substrate. They don't they are not structurally similar. They are not substrate analogs generally. The binding of them causes conformational change in the enzyme molecule, so they affect the affinity of the substrate binding, either increase it or decrease it. It is according to the type. If they are positive effectors, they enhance the rate, increase the affinity. If they are negative effector, they attenuate the rate, decrease the rate of the reaction. Okay, so allosteric effectors can be activator or positive effector, or can be inhibitors or negative effector. And if they are inhibitors, they are of the non-competitive type. Okay, allosteric effector affects the Km and the Vmax, both of them. And the binding of one, so promotes the effect on the binding of another. So the binding of the effector promote or has effect on the binding of substrate. So this is cooperativity. It can be positive cooperativity or it can be negative according to the influence. If it is positive effector, it has positive cooperativity. If it is negative, it has negative cooperativity. Covalent modification is regulation of an enzyme activity through covalent binding of a functional group. Okay, and this functional group commonly phosphate group. So sometimes known as phosphorylation, dephosphorylation. And the binding of the phosphate group is done by specific enzymes. Kinases facilitates the binding of the phosphate group, and phosphatases facilitates the removal of the phosphate group. So this is the enzyme. If it bound to a phosphate group, this is phosphorylated or covalent modified. If it releases this is dephosphorylated enzyme. The phosphorylation dephosphorylation is maintained by specific enzyme, kinases and phosphatases. Kinase add phosphate and phosphatase release the phosphate. Okay, and this process is under hormonal control. Some hormones facilitate phosphorylation by activation of kinases. Other hormones facilitate dephosphorylation by activation of phosphatases. Okay, and the addition of this phosphate group sometimes can enhance the activity of the enzyme, sometimes can depress the activity of the enzyme. There is no rule that phosphorylation increase or dephosphorylation decrease. Sometimes phosphorylation increase activity of enzymes, sometimes phosphorylation decrease the activity of enzymes. Sometimes dephosphorylation increase the activity of enzymes, sometimes dephosphorylation decrease the activity of enzymes. This depends on the enzyme itself. For example, glycogen synthase active with dephosphorylation, but glycogen phosphorylase active with phosphorylation. بالتالي العملية بتعتمد على الإنزيم نفسه. بعض الإنزيمات بتزيد بالفسفرة النشاط بتاعها، وبعضها بيقل بالفسفرة. فما يعني ما في يعني إنه الفسفر بيزيد النشاط بصورة عامة أو بتقلله بصورة عامة. There is another type of covalent modification. This is irreversible covalent modification. ده بيحصل لما يكون عندي enzyme released as inactive, then removal of some part of this enzyme makes this enzyme active. This is trimming or proteolytic cleavage. The inactive enzyme بيكون pro-enzyme or zymogen, and the removal of part of it is done by proteolytic cleavage, and this is irreversible process. So the enzyme become active. ودي بتحصل دايما في digestive enzymes، بتحصل في blood clotting factors. And the process is considered post-translational modification. After synthesis of the protein molecule, the trimming occur to make the enzyme active. Okay. Finally, we have some sort of feedback inhibition, that end product inhibition or feedback inhibition. End product inhibition is the product inhibits the enzyme itself. Feedback inhibition, one of the product in the pathway inhibits the enzyme itself. That is regarding the regulation of enzyme activity. Thank you.